Hi, welcome to ESI 521, Pattern Generation at the Nanoscale. I'm Professor Ukjun Nam at the Pennsylvania State University. So this lecture is the last lecture of unit number 4. And then we're gonna talk about DNA origami. So this is the lecture number 4 in unit 4. So here is the outline of today's lecture. So we're gonna talk about what is DNA origami, and then we will talk about DNA origami processes. So this is a little bit basics of the DNA. So as we discussed in the previous lectures at the molecular self-assembly, DNA is another kind of a good material which self-assembled based on these 4 bases. And then these 4 bases always kind of bind with specific base like this. And then this kind of a binding between the bases is happening by this kind of a hydrogen bonding. And right now I'm showing you double-stranded DNA, so we can just separate DNA. So if we just separate one single-stranded DNA has these are all backbones, and then it has an asymmetry end. So one end of the single-stranded DNA is an 3 prime and the other end is a 5 prime. So when those kind of single-stranded DNA combine each other, it has some kind of a preference of the end. And then, as I just briefly mentioned, it has this kind of 4 bases. So I will give you some I will show you some movies which give more introduction. So in this example, the movie will show you excuse me, how those kind of DNAs separated, and then even it can just replicate by itself. So we call that as a polymerase chain reaction. So based on this kind of replication capability of the DNA, we can just multiply the DNA. So this movie will show you more details about those kind of reactions.
Did you know that cells in your body have to regenerate and replenish themselves? Well, think about what happens to your skin in the winter. Your skin becomes dry and flakes off. What happens to those skin cells? Does your skin become so thin from loss of cells that you can see through it? Of course not. The cells regenerate to replace the ones that flaked off. This occurs through a process called mitosis. During mitosis, one cell divides to produce an identical copy of itself. If the cell is going to be identical, it must also have the same copy of DNA. DNA replication is the process where DNA makes an exact copy of itself for a new daughter cell just before mitosis. DNA replication is semi-conservative. This means that instead of having an entirely new copy of DNA for the new cell, a parent DNA strand splits in half, and each half adds a new complementary strand. The result is that each of the 2 double-stranded molecules of DNA are half old and half new. Let's explore how this happens. The first step in eukaryotic replication is for the enzyme helicase to unzip the double-stranded DNA molecule. The enzyme literally separates the 2 strands by breaking the hydrogen bonds between A and T and between G and C. Once helicase unzips the 2 strands of DNA, there are single-stranded binding proteins that hold the strands apart. An enzyme called primase can then put an RNA primer on the DNA molecule. The primer provides a free 3' OH so that a phosphodiester bond can be formed with the next nucleotide. DNA polymerase binds to and extends the sequence from the primer by adding complementary nucleotides. How does polymerase know which nucleotide to add? Well, if the original DNA molecule has an A, then polymerase will add a T. What do you think polymerase will add if the original molecule has a G? That's right, it'll add a C. It continues along the DNA molecule, adding the complementary nucleotide. Remember that a double-stranded DNA molecule is anti-parallel. This means if one strand is in the 5 to 3' direction, the complementary strand must be positioned in the 3 to 5' direction. Newly synthesized strands of DNA must be anti-parallel to the parent strand they are being copied from. However, the thing about DNA polymerase is that it only synthesizes new strands of DNA in one direction, only in the 5' to 3' direction. Because polymerase can only extend in the 5 to 3 direction, one strand follows along behind helicase and can extend continuously. This is called the leading strand. The other side has to wait for helicase to unzip another section of DNA so that it can lay down a primer and polymerase can extend. Because polymerase must wait for the replication fork to open, this strand is called the lagging strand and results in discontinuous fragments called Okazaki fragments, named after the scientist who discovered them. Last but not least, an enzyme called ligase comes along and seals the nicks between the Okazaki fragments to ensure there are no gaps in the phosphodiester backbone. DNA replication results in two identical copies of DNA that are half old and half new. The strands are complementary to one another. This new copy of DNA is ready to be placed in a new cell.
So this is the basics of the DNAs. So the reason why I showed you this video was first I just wanted to show you the basic structure of the DNA and then how those kind of DNAs, this kind of single-stranded DNAs can be engineered. Those kind of engineering DNA is kind of pretty important. So as we discussed with the other kind of technologies we talked about in Unit 4, like nanosphere lithography and then block polymer lithography, all those kind of common among those kind of technology is we do not need separate pictures like a photo mask or electronic file which containing some kind of a specific design, but instead of that we engineer this material, which is the medium, directly. So by applying some energies, we just wait this kind of engineered materials, self-assembled, and then just forming those kind of designed patterns we want to achieve finally. So for that engineering of the DNA, those kind of prime, and then those kind of bases, and then is kind of creation of those kind of synthesis of the other kind of base of the DNA is a very fundamental mechanisms using this DNA. So what is DNA origami? So using those kind of DNA, we talked about so far how we can make more complex structure. So just comparing this DNA origami compared with the other two techniques using molecular self-assembly like nanosphere lithography and then block polymer lithography, actually the DNA origami offers much more complex structures we can obtain. So this slide shows the example of the shape DNA origami can offer. These are all two-dimensional design. So we can make square, we can make a rectangle, and then we can make a star, smiley face, and this kind of triangular structure. And then as you can see, this dimension, whole dimension of the box is only 100 nanometer. So as to the dimension control, this is as fine as those kind of most of the advanced lithography can offer. So this first row shows the computer design to achieve or this kind of a DNA origami structure. And then this third and second and third row shows the actual image of the DNA origami result, imaged by the atomic force microscope. So here we're gonna talk about a little bit more about how we can engineer further this DNA to make this kind of a specific all different kind of shapes of shapes. So here is the movie explaining that.
So people argue vigorously about the definition of life. They ask if it should have reproduction in it, or metabolism, or evolution. And I don't know the answer to that, so I'm not going to tell you. I will say that life involves computation. So this is a computer program. Booted up in a cell, the program would execute, and it could result in this person. Or with a small change, it could result in this person, or another small change, this person, or with a larger change, this dog, or this tree, or this whale. So now if you take this metaphor as genome as program seriously, you have to consider that Chris Anderson is a computer fabricated artifact, as is Jim Watson, Craig Venter, as are all of us. And in convincing yourself that this metaphor is true, there are lots of similarities between genetic programs and computer programs that could help convince you. But one to me that's most compelling is a peculiar sensitivity to small changes that can make large changes in biological development. The output, a small mutation can take a 2-wing fly and make it a 4-wing fly. Or it could take a fly and put legs where its antenna should be, or if you're familiar with The Princess Bride, it can create a 6-fingered man. Now, a hallmark of computer programs is just this kind of sensitivity to small changes. If your bank account's $1 and you flip a single bit, you can end up with $1,000. So these small changes are things that I think that indicate to us that a complicated computation in development is underlying these amplified large changes. So now, all of this indicates that there are molecular programs underlying biology, and it shows the power of molecular programs, biology does, and what I want to do is write molecular programs potentially to build technology. And there are a lot of people doing this, a lot of synthetic biologists doing this, like Craig Venter, and they concentrate on using cells. They're cell-oriented. So my friends, molecular programmers, and I have a sort of biomolecule-centric approach. We're interested in using DNA, RNA, and protein and building new languages for building things from the bottom up, using biomolecules potentially having nothing to do with biology. So these are all the machines in a cell. There's a camera, there's the solar panels of the cell, some switches that turn your genes on and off, the girders of the cell, motors that move your muscles. My little group of molecular programmers are trying to refashion all of these parts from DNA. We're not DNA zealots, but DNA is the cheapest, easiest to understand, and easy to program material to do this. And as other things become easier to use, maybe protein will work with those. If we succeed, what will molecular programming look like? You're going to sit in front of your computer, you're going to design something like a cell phone, and in a high-level language, you'll describe that cell phone. Then you're going to have a compiler that's going to take that description, and it's going to turn it into actual molecules that can be sent to a synthesizer, and that synthesizer will pack those molecules into a seed. And what happens if you water and feed that seed appropriately is that it'll do a developmental computation, a molecular computation, and it'll build an electronic computer. And if I haven't revealed my prejudices already, I think that life has been about molecular computers, building electrochemical computers, building electronic computers, which together with electrochemical computers will build new molecular computers, which will build new electronic computers, and so forth. And if you buy all of this and you think life is about computation as I do, then you look at big questions through the eyes of a computer scientist. So one big question is, how does baby know when to stop growing? And for molecular programming, the question is, how does your cell phone know when to stop growing? Or how does a computer program know when to stop running? Or more to the point, how do you know if a program will ever stop? There are other questions like this too. One of them is Craig Venter's question. Turns out I think he's actually a computer scientist. He asks, how big is the minimal genome that'll give me a functioning microorganism? How few genes can I use? This is exactly analogous to the question, what's the smallest program I can write that'll act exactly like Microsoft Word? And just as he's writing, bacteria that it will be smaller, he's writing genomes that'll work, we could write smaller programs that would do what Microsoft Word does. But for molecular programming, our question is, how many molecules do we need to put in that seed to get a cell phone? What's the smallest number we can get away with? Now, these are big questions in computer science. These are all complexity questions. And computer science tells us that these are very hard questions. Almost many of them are impossible. But for some tasks, we can start to answer them. So I'm going to start asking those questions for the DNA structures I'm going to talk about next. So this is normal DNA. What you think of it as normal DNA. It's double-stranded. It's a double helix. It has the A's, T's, C's, and G's that pair to hold the strands together. And I'm going to draw it like this sometimes, just so I don't scare you. We want to look at individual strands and not think about the double helix. When we synthesize it, it comes single-stranded. So we can take the blue strand in one tube and make an orange strand in the other tube, and they're floppy when they're single-stranded. You mix them together, and they make a rigid double helix. Now, for the last 25 years, Ned Seaman and a bunch of his descendants have worked very hard and made beautiful three-dimensional structures using this kind of reaction of DNA strands coming together, but a lot of their approaches, though elegant, take a long time. They can take a couple years or can be difficult to design. So I came up with a new method a couple years ago I call DNA origami that's so easy you could do it at home in your kitchen and design the stuff on a laptop. But to do it, you need a long single strand of DNA, which is technically very difficult to get. So you can go to a natural source. You can look in this computer-fabricated artifact, and he's got a double-stranded genome that's no good. You look in his intestines, there are billions of bacteria. They're no good either, double-strand again. But inside them, they're infected with a virus that has a nice, long, single-stranded genome that we can fold like a piece of paper. And here's how we do it. This is part of that genome. We add a bunch of short synthetic DNAs that I call staples. Each one has a left half that binds the long strand in one place, and a right half that binds it in a different place, and brings the long strand together like this. The net action of many of these on that long strand is to fold it into something like a rectangle. Now, we can't actually take a movie of this process, but Sean Douglas at Harvard has made a nice visualization for us that begins with a long strand and has some short strands in it. And what happens is that we mix these strands together, we heat them up, we've got a little bit of salt, heat them up to almost boiling and cool them down. And as we cool them down, the short strands bind the long strands and start to form structure. And you can see a little bit of double helix forming there. When you look at DNA origami, you can see that what it really is, even though you think it's complicated, is a bunch of double helices that are parallel to each other. And they're held together by places where short strands go along one helix and then jump to another one. So there's a strand that goes like this, goes along one helix and binds and jumps to another helix and comes back that holds the long strand like this. Now, to show that we could make any shape or pattern we wanted, I tried to make this shape. I wanted to fold DNA into something that goes up over the eye, down the nose, up the nose, over around the forehead, back down, and end in a little loop like this. And so I thought this could work, anything could work, so I had a computer program design the short staples to do this. I ordered them, they came by FedEx. I mixed them up, heated them, cooled them down, and I got 50 billion little smiley faces floating around in a single drop of water. And each one of these is just 1,000th, they're 1,000th the width of a human hair, okay? So they're all floating around in solution. And to look at them, you have to get them on a surface where they stick. So you pour them out onto a surface, and they start to stick to that surface, and we take a picture using an atomic force microscope that's got a needle, like a record needle, that goes back and forth over the surface, bumps up and down, and feels the height of the first surface. It feels the DNA origami. There's the atomic force microscope working. And you can see that the landing's a little rough when you zoom in. They've got, they have weak jaws that flip over their head, and some of their noses get punched out, but it's pretty good. You can zoom in and even see the extra little loop as little nano goatee. Now, what's great about this is anybody can do this. And so I got this in the mail about a year after I did this, unsolicited. Anyone know what this is? What is it? It's China, right? So what happened is a graduate student in China, Lulu Xian, did a great job. She wrote all her own software to design and build this DNA origami, a beautiful rendition of China, which even has Taiwan, and you can see that it's sort of on the world's shortest leash, right? So this works really well, and you can make patterns as well as shapes, okay? And you can make a map of the Americas and spell DNA with DNA. And what's really neat about it, well, actually this all looks like nano artwork, but it turns out that nano artwork is just what you need to make nano circuits. So you can put circuit components on the staples, like a light bulb and a light switch, let the thing assemble, and you'll get some kind of a circuit. And then you can maybe wash the DNA away and have the circuit left over. So This is what some colleagues of mine at Caltech did. They took a DNA origami, organized some carbon nanotubes, made a little switch, you see here, wired it up, tested it, and showed that it is indeed a switch. Now, this is just a single switch, and you need 500 million for a computer, so we have a long way to go, but this is very promising because the origami can organize parts just 1/10 the size of those in a normal computer, so it's very promising for making small computers. Now, I want to get back to that compiler. The DNA origami is a proof that that compiler actually works. So you start with something in the computer, you get a high-level description of the computer program, a high-level description of the origami. You can compile it to molecules, send it to a synthesizer, and it actually works. And it turns out that a company has made a nice program that's much better than my code, which is kind of ugly, and will allow us to do this in a nice visual computer-aided design way. So now you can say, all right, why isn't DNA origami the end of the story? You have your molecular compiler, you can do whatever you want. The fact is that it does not scale. So if you want to build a human from DNA origami, the problem is you need a long strand that's 10 trillion bases long. That's 3 light years worth of DNA. So we're not going to do this. We're going to turn to another technology called algorithmic self-assembly of tiles. It was started by Eric Wintry. And what it does is it has tiles that are 1/100th the size of a DNA origami. You zoom in, they're just 4 DNA strands, and they have little single-stranded bits on them that can bind to other tiles if they match. And we'd like to draw these tiles as little squares. And if you look at their sticky ends, these little DNA bits, you can see that they actually form a checkerboard pattern. So these tiles would make a complicated self-assembling checkerboard. And the point of this, if you didn't catch that, is that tiles are a kind of molecular program, and they can output patterns. And a really amazing part of this is that any computer program can be translated into one of these tile programs, specifically counting. So you can come up with a set of tiles that when they come together form a little binary counter rather than a checkerboard. So you can read off binary numbers 5, 6, and 7. And in order to get these kind of computations started right, you could need some kind of input, a kind of seed. You could use DNA origami for that. You can encode the number 32 in the right-hand side of a DNA origami. And when you add those tiles that count, they will start to count. They'll read that 32 and they'll stop at 32. So what we've done is we figured out a way to have a molecular program know when to stop going. It knows when to stop growing because it can count. It knows how big it is. So that answers that sort of first question I was talking about. It doesn't tell us how babies do it, however. So now we can use this counting to try and get at much bigger things than DNA origami could otherwise. Here's the DNA origami, and what we can do is we can write 32 on both edges of the DNA origami, and we can now use our watering can and water with tiles, and we can start growing tiles off of that and create a square. The counters serve as a template to fill in a square in the middle of this thing. So what we've done is we've succeeded in making something much bigger than a DNA origami by combining DNA origami with tiles. And the neat thing about it is that it's also reprogrammable. You can just change a couple of the DNA strands in this binary representation, and you'll get 96 rather than 32. And if you do that, the origami is the same size, but the resulting square that you get is 3 times bigger. So this sort of recapitulates what I was telling you about development. You have a very sensitive computer program where small changes, single tiny little mutations, can take something that made 1 size square and make something very much bigger. Now, this, using counting to compute and build these kinds of things by this kind of developmental process is something that also has bearing on Craig Venter's question. So you can ask how many DNA strands are required to build a square of a given size. If we wanted to make a square of size 10, 100, or 1,000, if we use DNA origami alone, we would require a number of DNA strands that's the square of the size of that square. So we need 100, 10,000, or 1,000,000 DNA strands. That's really not affordable. But if we use a little computation, we use origami plus some tiles that count, then we can get away with using 100, 200, or 300 DNA strands. And so we can exponentially reduce the number of DNA strands we use if we use counting, when we use a little bit of computation. And so computation is some very powerful way to reduce the number of molecules you need to build something, to reduce the size of the genome that you're building. And finally, I'm going to get back to that sort of crazy idea about computers building computers. If you look at the square that you build with the origami and some counters growing off it, the pattern that it has is exactly the pattern that you need to make a memory. So if you affix some wires and switches to those tiles rather than to the staple strands, you affix them to the tiles, then they'll self-assemble the somewhat complicated circuits, the demultiplexer circuits you need to address this memory. So you can actually make a complicated circuit using a little bit of computation. It's a molecular computer building an electronic computer. Now you'll ask me, how far have we gotten down this path? Experimentally, this is what we've done in the last year. Here's a DNA origami rectangle, and here's some tiles growing from it, and you can see how they count. 1, 2, 3, 4, 5, 6, 9, 10, 11, 12, 17. So it's got some errors, but at least it counts up. So it turns out we actually had this idea 9 years ago, and that's about the time constant for how long it takes to do these kinds of things. So I think we've made a lot of progress. We've got ideas about how to fix these errors, and I think in the next 5 or 10 years we'll make the kind of squares that I described, and maybe even get to some of those self-assembled circuits. So now, what do I want you to take away from this talk? I want you to remember that to create life's very diverse and complex forms, life uses computation to do that. And the computations that it uses, they're molecular computations. And in order to understand this and get a better handle on it, as Feynman said, we need to build something to understand it. And so we are going to use molecules and refashion this thing, rebuild everything from the bottom up, using DNA in ways that nature never intended, using DNA origami and DNA origami to seed this algorithmic self-assembly. So this is all very cool, but what I'd like you to take from the talk, hopefully from some of those big questions, is that this molecular programming isn't just about making gadgets. It's not just making self-assembled cell phones and circuits. What it's really about is taking computer science and looking at big questions in a new light, asking new versions of those big questions, and trying to understand how biology can make such amazing things. Thank you.
So this movie shows you very basics and then very informative information about how people can design DNA and how those kind of a designed engineered DNA can self-assembly to make this kind of a specific shape. And even this video go one step further and then just talked about using this kind of a two-dimensional DNA origami, how we can potentially use this kind of DNA patterns for potential use. And then the example is just a computational application. So basically, what we wanted to do to make this kind of a two-dimensional structure is using the computer, we can just design one long single-stranded DNAs and then couple of the short DNA which work as stapler. So as those kind of DNAs just heated up and then cooling down, you can just making and then specific bindings based on our design. You can just make this kind of a specific structure. So we only talked about two-dimensional DNA origami, but we can make three-dimensional DNA origami as well. So if we just make three-dimensional structures using this kind of same kind of principles, we can just make this kind of three-dimensional structures. But the basic idea of making this kind of three-dimensional structure is kind of very similar to this. So actually the videos that I showed you is not made of DNA, and then actually you might have a chance to see such a toys in a kind of a toy store. Actually that is an magnet and then those kind of metal rod. So using that way people can just make more complex 3D-dimensional structures. The DNA origami for the three-dimensional structures, the basic idea is kind of very similar to playing with that kind of a toy. So this structure is kind of very similar to the structures we saw made from that toy. So actually all these kind of three-dimensional structures can be made out of this kind of three-dimensional DNA origami. So if we are making these kind of structures, we can just make these kind of structures and then these kind of cores for the microfluidic or nanofluidic application. So this is a more likely bottom-up approaches. So if we making this kind of three-dimensional structures directly using the DNA origami, it is more likely rather than it is more likely a three-dimensional kind of bottom-up approaches fabricating some structures. So the way of we are making this kind of three-dimensional structure is kind of very close to the craft using straw. So using straw, people can make hats and those kind of a craft. So the basic idea is the same. So using those kind of a DNA origami, we can just make this kind of a twisted bundles of these rod structures. So using this kind of DNAs, we will make a bunch of these rod-like structures. And then we also use this kind of small stapler functioning DNA. So this rod structure of the DNAs is folded like this kind of strategies. So as it just folded like this, we can just make this kind of a cube structure. This is this kind of a structure. So this is the basic idea for creating three-dimensional structures out of the DNA origami. But as to the process itself, it is a little bit complex for designing, but the basic process is very similar to the process needed for the two-dimensional structure formation. And this is the basic design people want to achieve. And then all these pictures are actual pictures based on those kind of a DNA origami. So as you can see, this kind of resulting electronic microscope images just matches exactly from what we designed from computer. And then all these kind of structures based on this kind of different kind of folding times, and then there is some kind of select the selection of the lengths of the DNAs or things like that. So that is the another kind of a way we needed to do this kind of assembly. But basically, using this kind of long DNAs with this kind of a shape, as this kind of branches, just folding each other like this, just combining all these three structures, we can just make these kind of structures as well. And then these are all the actual images of the result. And as we just go more kind of a complex design, we can make this kind of a bar structures, and then this kind of a twisted bar structures, and then we make this kind of a U or V shaped or this kind of a much more complex structures like a gear structure. So for the secret of that is based on the base pair, number of the base pair, we can just make the road structures to be stranded one directions to the other, and then we can also make these structures to be this kind of a curved structure as well, and that is all depends on the design of the number of the base pair. So using that kind of a basic idea, we can just make this kind of a flat bar structures, and we can just make this kind of a twisted bar structures as well, and then twisted another direction, and then this is an actual result. And then putting some other kind of a DNAs in the middles, and then based on the same kind of ideas, we can just make a straight bar, or we can just control the angle of the bar like this from 0 degree to the all the way down to the 180 degree bend structure, and then these are all the result. So when we properly design these DNAs, we can even make this kind of a much more complex structures, and then which is the gear-like structure. So if you are just thinking about making this kind of a gear-like structures using traditional way, it takes much more kind of processes, like it needs an lithography process, which needs a very high resolution, and it also needs an deposition process to make this kind of a film deposition, and then it also needs an etching process to remove unnecessary materials to form this kind of a structure. So in a sense, this DNA origami is it belongs to the bottom-up approaches, and then designing, engineering this kind of DNA, we can just easily achieve this kind of a very complex structures. And this is the kind of a program. This is a free software you can download from this website. So if you are interested in designing or further exploring the possibility of this kind of design of the DNA origami, you can just stop by this website and then download the software, and then you can try, and then you can make your own structures based on this basic idea of the DNA origami. So we talked about the DNA origami in this lecture, and then this is another kind of lithography technology or pattern transferring technology using molecular self-assembly. So just simply comparing with the other kind of two technologies like nanosphere lithography or block polymer lithography, actually the nanosphere lithography only offers the dot patterns, which is hexagonally arranged, and then the block polymer offers a little bit more kind of lamellate structures and this kind of hexagonal structures, and then it also offers this kind of body centered cubic structures. But these two methods has a limitation in terms of the designs we can achieve. But as you saw from the examples, this DNA offers much more variety of the pattern formation. So if you are designing the DNA properly, even it can just make some maps, like in Chinese kind of maps. So in terms of the pattern definition, this technology is much more flexible compared with the other two methods. And then properly engineered DNAs not only make this kind of a two-dimensional patterning, but also it can also directly build three-dimensional structures. So this is the last lecture for the Unit 4. So we will talk about the last unit, which is on additive manufacturing. So in the last unit, we're going to talk about 3D printing, which is one of the technologies used for the additive manufacturing. So thank you, and then see you next time.